Hey, thank you for that introduction. My name is Ryan Baumgarten, as I was just introduced. First, I'd like to begin with an overview of today's presentation. I will be presenting a brief introduction to some of the challenges that are faced by proteomic researchers and biologists using mass spectrometry to study complex protein samples. Then my colleague, Barb Kabord, will present on different strategies to overcome some of these challenges. Three of the strategies that we will cover in depth include subcellular protein fractionation, integral plasma and organelle membrane protein enrichment, and immunoprecipitation mass spectrometry, or IPMS, which is a powerful immunoselection technique for targeted mass spectrometry applications. One of the major challenges faced by biologists doing mass spectrometry-based proteomics is that a majority of the proteomics researchers are focused on protein identification and characterization. As such, the instruments and methods available for proteomic analyses using mass spectrometry can most readily provide information about a protein sequence and its various isoforms, including a wide variety of post-translational modifications. Traditional methods used to prepare protein samples for MS analysis requires enzymatic digestion of proteins to peptides. This approach, commonly referred to as bottom-up proteomics, relies on peptides for database searching and subsequent protein identification. However, much of the original protein biological context is lost during this type of sample preparation. Proteins are typically extracted from whole cell lysates using denaturing buffers, which results in loss of information regarding protein localization and protein-protein interactions. Although protein expression data can be inferred from quantifying peptides, this information alone is not sufficient to understand the biological pathways that are active in a cell. Another challenge faced by biologists doing mass spectrometry experiments is that the list of proteins identified by global discovery proteomics is not complete. An example of how this is shown on the left, where a typical discovery proteomic dataset can readily identify and quantify thousands of proteins across multiple sample types or conditions. However, when mapped on the known biological signaling pathways, many of the known lower abundant proteins are not detected. In addition, many of the PTMs that have known function in the literature may not be identified by MS due to low protein sequence coverage. This lack of comprehensiveness results in major gaps and provide an incomplete analysis of the proteome, making it difficult to test hypotheses regarding specific proteins or to validate data observed with immunoassays such as Western blot or ELISA.